I'm R.K. Walker here at NASDAQ MarketSite with a Financial Buzz exclusive interview. Alongside me today is Jim Joyce, CEO and founder of Aethlon Medical Inc. Welcome, Jim.
RK, pleasure to be here.
Why don't you tell us a little bit about your company?
Well, the basis of our company is the development of unique and novel affinity biofiltration devices that can address life-threatening disease conditions.
And it's my understanding your flagship product is the Hemo Purifier.
That's correct. It's a product that has an affinity to have broad spectrum activity against infectious viral pathogens. And it has a unique methodology of being able to capture viral pathogens by a structure that they co-opt from your own host cells during replication. It's called a glycan shield. And we attach and eliminate on a very rapid basis a wide range of viruses from circulation.
In layman's terms, what does that do?
It provides a very rapid elimination of virus from circulation as well as shed toxins that suppress the immune system. For acute viral pathogens like Ebola, Zika virus, things of that nature, we have a mechanism that can quickly tip the balance in favor of the immune system. And this is really, quite an urgent requirement because traditional drugs and vaccines just can't be developed to address each and every threat that evolves.
How does it go about this? You hook it into your bloodstream?
Yeah, the device works on the global infrastructure of dialysis, CRRT machines, already located in hospitals and clinics worldwide. The device fits in that infrastructure. We access the blood circulatory system. The blood flows through the cartridge about once every 15 minutes, and it has a selective affinity to capture viruses and shed glycoproteins. And it's worked very well in human treatment experiences.
You mentioned Zika virus.
Yes.
So any of these viruses it can be applied to, or are you still in a testing phase?
Well, we've had the very good fortune of working with leading government and non-government research institutes to validate the capture of a wide range of pathogens, Ebola, West Nile virus, multiple strains of pandemic influenza virus, chikungunya virus, dengue virus, and then leveraged those outcomes into human studies where we've treated HIV/AIDS patients, hepatitis C patients, and recently got a lot of attention for the successful treatment of a comatose Ebola patient who made a remarkable recovery.
Right, that was, you were awarded in 2014 Time Magazine for a new invention.
Top invention of 2014, as well as a remarkable advance in healthcare. Ironic that we've been working on this for a number of years. It took us till 2014 to receive such an award.
Right, and this is rather unique in that it doesn't just filter out everything, it filters out this specific virus.
Okay, that's a very good point because historically people have tried to use biofiltration devices to eliminate toxins and other deleterious factors from the circulatory system. Generally, those mechanisms have been with hollow beads or hollow fibers to remove things solely by molecule size. In our case, we have a mechanism to remove and segregate viruses away from the bloodstream, away from blood cells, and then selectively capture them through the immobilization of an agent that binds to that unique glycan shield. And that shield is very important. It's common amongst all viruses, and it's the mechanism viruses use to help evade detection of the immune system, and we're using that structure actually against the viruses.
Can it be used by itself as a sole treatment, or is it intended to be used with other current existing treatments?
We look at this as a means in an acute viral pathogen to tip the balance in favor of the immune system. In chronic conditions like hepatitis C or HIV, it's a means to complement. Technologies are advancing antiviral agents where there's an evolution of drug resistance, a means to kind of reset viral load or to strip out mutant viral strains that are causing the patients to fail their antiviral drug therapies.
Are there any future applications that you're working on that you haven't discussed yet?
Well, the thing that we're extremely excited about, and we actually started this research in 2007, we discovered that particles secreted by tumors in cancer called tumor-secreted exosomes. We believe these particles were very immunosuppressive, much like the proteins that shed from the surface of viruses after replication. However, at the time, these particles were deemed by the medical community to be cellular debris with no biological function. But we discovered that the affinity lectin we immobilize in our Hemo Purifier that allows us to capture viruses is effective in binding tumor-secreted exosomes, which are approximately the same size as viruses. Today, tumor-secreted exosomes have suddenly become a very important target in cancer care. They're not only directly immunosuppressive to the immune cells you need to combat cancer, but they have been discovered to be the seeds that allow the creation and spread of metastases in cancer patients. So this is an exciting area for us as a company that we're pursuing as well, in addition to the infectious viral pathogen opportunity. And we believe that a device that could remove these targets to eliminate the immune suppression, to slow down the spread of metastases, really are the other side of the equation to emerging immuno-oncology drugs, which are designed to stimulate the immune response. So we think a device that can fit well with those drugs without adding additional drug toxicity could play a very important factor in the future of cancer care.
Right, so they actually filter out the exosomes, is that it?
That's correct.
That are related to the cancer, so it makes the treatments more effective.
That is the objective. What we find fascinating is that it seems to be that tumor-secreted exosomes and viruses are sharing the same evolutionary mechanism to evade surveillance by the immune system by coating themselves with this unique structure that's pulled from it's co-opted from the host itself. And the host's immune system can't respond. It can't tell the difference between self and non-self. And that's the structure we're looking at. So we find it very intriguing that such a structure would exist both in oncology, in cancer, as well as infectious disease related to viral pathogens.
Interesting. You also have a contract with DARPA right now. I believe you're studying sepsis.
That's correct.
Why don't you elaborate on that?
Sure. We are in year 5 of a 5-year contract. And the goal of this program with DARPA, it's called the Dialysis-Like Therapeutics Program, and we have a team of great researchers from different organizations. The goal of the program is to develop an extracorporeal device to treat sepsis, which is a leading cause of death in wounded warfighters as well as in the ICU and civilian populations. Our objective in the program is to take our affinity biofiltration mechanisms and adapt them towards sepsis. We're very excited about the program. To date, we've achieved more than 25 different milestones under the program, with milestone payments of approximately $5.4 million to the company. And we hope to be able to expand our opportunities beyond infectious disease and our hope in oncology to include sepsis at some point in time in the future as well.
You've recently uplisted to NASDAQ.
That's correct.
How is that going for you?
We are enjoying the benefit of being on NASDAQ. We uplisted in June. We really worked hard to survive for many years as a public company on the over-the-counter marketplace. Based on different changes in the marketplace, it really got to the point where most institutional investors were precluded from participating in public companies that didn't trade on a major exchange like the NASDAQ. So uplisting to NASDAQ has increased our ownership profile to include institutional investors. We actually had a filing this week of a new institutional investor that's taken a 9.8% ownership in the company.
Now that you have a higher profile, what are your short-term goals over the next, say, 2-3 years?
Well, the short-term goals are to execute on the foundation that we've put in place over the last decade. You know, it took us a long time to uplist the NASDAQ, but we stuck to our guns and we didn't change our programs. We focused. We continued to advance each and every year. We clinically advanced our technology towards marketplace. We're now in FDA-approved clinical studies, and we need to execute on our clinical progression model here in the U.S. We also need to leverage what we've learned over the last decade in creating the hemopurifier to be able to leverage opportunities to create new affinity biofiltration devices that can address other life-threatening disease conditions where targets that lead to disease progression are known to be in circulation. And we're in discussions with different groups about how we might do that related to disease conditions we haven't discussed before.
You're a majority shareholder of Exosome Sciences, correct?
We are.
What are you doing with Exosome right now?
Well, that's a very interesting scenario. Exosome Sciences is a diagnostic company of which we don't have a core competency, quite honestly, in diagnostics, but what we understood based on our original work and learning how to create affinity techniques to bind tumor-secreted exosomes, we envisioned that the intellectual property that was issued for those capabilities also taught methodologies to isolate particles that would be the basis of diagnostic techniques. And so exosomes have evolved to become an important biomarker underlying cancer and other disease conditions. And we were very interested in chronic traumatic encephalopathy. We had a very close relationship with the Boston University CTE Center, who's well known for their work in the field. And we got invited to participate in looking for a biomarker for CTE, which is currently diagnosed at autopsy after death of an individual.
And it's primarily caused by repetitive head trauma, correct?
Repetitive subconcussive blows. That's correct. A lot of people equate it with traumatic brain injury or concussions, but really the commonality for people that have been diagnosed with CTE is they participated in sports, primarily in the NFL, in sports or football that created or activities where there was repetitive subconcussive blows to the individuals. So there's a lot of interest for developing a biomarker that could identify CTE in individuals that are living. So we got invited to participate in the DETECT study. Our exosomal tau biomarker that we discovered, we trademarked the name Tauzome, was being tested as a candidate biomarker. We saw that in former NFL players, the levels of this biomarker are significantly higher in former NFL players as compared to non-NFL controls. And we also saw that the levels of cognitive decline seem to associate with Tauzome levels in these former NFL players. So today we have a manuscript out for publication that's been accepted, so I expect in the next 30 to 60 days a scientific journal will publish the hard data from our results. We've agreed to continue testing of former NFL players that participated in the DETECT study, to continue to grow a database and looking at changes of their Tauzome levels going into the future. And we're very excited about a $16 million NIH grant that's just been awarded to our collaborators, where they'll continue to advance our Tauzome testing programs in a much larger recruitment of former NFL players at up to 17 different clinical sites. So that's a very exciting program for us, and our hope, again, we're not detecting concussions, not traumatic brain injury, but a marker that can identify and monitor changes in Tauzome levels, which we believe might be a biomarker for identifying CTE in living individuals.
Right. Earlier you mentioned you've been doing this a while. Any reason you want to elaborate briefly why now is a particularly good time to take another look at your company?
Well, I think the one thing we've never done is we've never been pushed to change where we were taking the company. So even in times where it's been suggested maybe we should change course, we never change course. We believe that in the future that global warming, urban crowding, the change of vector evolution, how viruses can be transmitted, we believe that these conditions were going to become more and more prevalent. And we don't think, and we didn't think at the time, that it would be possible to align traditional antiviral drug or vaccine strategies with each and every threat. We believe very strongly there had to be a broad spectrum mechanism, not just against newly emerging threats, but most experts would tell you that it's inevitable that new genetically modified threats to defeat antiviral drugs could be released as agents of bioterror. And at the rates of mutation of new emerging threats, we're going to continue to see the release through Mother Nature of new threats that evolve, that are unknown today, that are going to be infectious to man. There are hundreds of viruses people don't recognize that are infectious to man, but fewer than 10 are addressed with antiviral drug agents.
Thank you very much, Jim. On that note, we'll conclude things for today.
RK, it's a pleasure. Thanks for having me on.
I'd like to thank again our guest, Jim Joyce, CEO and founder of Aethlon Medical. I'm RK Walker. Thank you for joining us, and I'll see you again next time.